I am I'm based at the University of Otago in New Zealand. So I'm sure all of you have heard of New Zealand. If we were doing, an in-person seminar, I'd maybe bring somebody up and ask them to put a pin on the map of exactly where New Zealand is, and you'd maybe choose here or here or here. But as we can't do that interactively, I'll just show you that New Zealand is here. So it's a couple of thousand miles off the coast of Australia. It has a population of 5.2 million people. It's smaller in population than the greater Atlanta area. So I'm based at the University of Otago, which is here in a little town called Dunedin. And then the other main cities are Auckland and Wellington, and we've also got Christchurch. So just to give you some background on the research I'm doing and how I've got to where I am, I moved to the University of Otago about 6 or 7 years ago. I was at the University of Michigan before that, and I've been, prior to moving to the University of Otago, interested in how reproductive processes can have long-term effects on metabolism and aging, really from kind of like a basic biology discovery science perspective, not really thinking about any kind of clinical outcomes or translational approaches. And when I moved to New Zealand, I became aware that there's a big push to do research that's going to have an impact for New Zealand. We don't have very much money, particularly if you want to get research funding from the Health Research Council, so similar to the NIH. You really need to be showing that your research is working towards some kind of real impact to the New Zealand population and something that's of importance to the New Zealand population. The other thing that I'll talk about later on in the talk is that we're also expected and should be working towards doing research that's of importance to the Indigenous people of New Zealand, which is the Maori community. So New Zealand has a legal treaty to conduct health research and to have a health system that provides equity for the Indigenous people of New Zealand, Maori people, to have equity in health outcomes compared to New Zealand European people. So when we're thinking about our research projects, we need to think about how they are going to lead to equitable health outcomes. And so when I moved to New Zealand, I actually met predominantly at first with several prominent Maori academics at the University of Otago, and I talked to them about my research program about how reproduction influences aging, and they were interested in that, but they actually said, what we really care about is the diseases that occur specifically during pregnancy, which disproportionately affect our community. And gestational diabetes was specifically raised to me by several women. So they said, there's this perception within the Maori community that gestational diabetes might almost be an inevitable part of pregnancy. So research in this area would be perceived as important. So over the last few years, I've been trying to kind of switch some of my research focus to applying the skills I have from kind of preclinical research using animal models to address questions that might then have translational outcomes. So I'll give you some background on gestational diabetes, which is where we have been switching our focus. So gestational diabetes mellitus is a form of diabetes that arises specifically during pregnancy. So during pregnancy, the baby and the placenta produce hormones that influence the mother's physiology. So things like placental lactogens, growth hormones, and other hormones and substances. And these act on maternal physiology to reduce the mother's insulin sensitivity. So this happens in all pregnancies. This helps to raise glucose levels and transfer those nutrients to help the baby's growth. But this causes, as I said, an impairment in maternal insulin sensitivity. And so during pregnancy, the mother's pancreas adapts, shows changes in beta cell number and beta cell function. This has been shown in humans and in mice, and leads to mothers producing more insulin. So this helps to combat that impairment. So in most pregnancies, these kind of co-adaptions lead to mothers being able to maintain their energy levels, control their blood glucose during pregnancy. But in some pregnancies, something goes wrong in this co-adaptions and a diabetic state occurs. So we have uncontrolled glucose circulating the body, and we have this diabetic state. This could be because of impairments in insulin secretion, or it could be due to impairments in insulin sensitivity. There are various different kind of diagnostic criteria for gestational diabetes that vary across the globe, but we could say that perhaps 10% of pregnancies in approximately 10% of pregnancies, gestational diabetes can occur. So gestational diabetes can lead to an increased risk of large babies at birth, macrosomia. It also increases the risk of preterm birth and other pregnancy conditions such as preeclampsia. When the baby is born, this diabetic state typically resolves. But it still leaves the mother with an increased risk of type 2 diabetes and cardiovascular disease. So I think there's a sevenfold increased risk of the mother getting type 2 diabetes compared to the general population after experiencing gestational diabetes. In addition, children born from mothers who've had one of these diabetic pregnancies can also have an increased risk of type 2 diabetes and cardiovascular disease. So this is a disease that arises during pregnancy, which has important and deleterious health outcomes for both the mother and the child. So when I started thinking about gestational diabetes and potential factors that influence gestational diabetes risk and that might be modifiable within populations in New Zealand, I came across a lot of research, human epidemiological research that had looked at the link between maternal diabetes and gestational diabetes risk. So there have been a number of different observational studies that have looked at maternal diet and the effects of maternal diet on gestational diabetes risk. So a number of different studies had found that mothers who eat high levels of protein have an increased risk of gestational diabetes. However, other studies have found a link to high fat intake and increased gestational diabetes risk. Although when mothers are eating high fat, because this is a calorific molecule, and they're also having high levels of energy intake too. And then there are also other studies that have linked high sugar intake to increased gestational diabetes risk. So there was a lot of evidence that maternal diet can influence gestational diabetes risk, but it's unclear for exactly what the causal roles of specific macronutrients might be within this process. So obviously, all of the foods we eat vary in the amount of fat, protein, and carbohydrates that they have. These are the macronutrients I'm referring to. And, if somebody's eating high levels of protein, this has been associated with gestational diabetes. But then they may also be eating other things in association with that high protein that haven't been recorded that could lead to these relationships. Also, kind of dietary recall and dietary food frequency questionnaires are also quite inaccurate. So this research has still kind of left us with a relatively poor understanding of actually which macronutrients causally influence gestational diabetes risk. So just to give you an idea of the strength of some of the relationships seen in these studies, last year there was a meta-analysis published looking at the relationship between protein intake and gestational diabetes. So they looked at various observational studies that have looked at the relationship between protein intake and gestational diabetes. So here is the forest plot from these individual studies looking at protein intake during pregnancy or protein intake pre-pregnancy. And basically what they find is that high protein intake, so protein intake, which is about 20% of daily energy intake, compared to low protein intake, so where protein intake is about 11% of total energy intake. This high intake is associated with a doubled risk of gestational diabetes. So, this is a substantial increase in risk. And what I would highlight is that this level of protein intake, that's actually within the recommended or the acceptable range recommended by various different health organizations. So this is within the normal range of protein intake. We're seeing this increased risk of gestational diabetes. So we wanted to try to conduct some preclinical research to see whether intake of different macronutrients might influence metabolic health during pregnancy. And we decided to do some experiments using an approach called nutritional geometry, using mice as a model. So this sort of preclinical testing process was pioneered by a group in Australia, where they've developed a range of diets that vary systematically in the amount of energy they get from different macronutrients. So they've designed 10 diets that all have the same energy density. So they provide the same amount of energy, the same number of calories per gram of food, but they differ systematically in the amount of protein, carbohydrate, and fat that they have. So we've got diets that we can use and apply to mice, which range in the amount of protein they have as a contribution to total energy intake, going from 7% of energy from protein, which is really quite a low level of protein. This is sort of undernutrition, all the way up to 50% of their energy from protein. So this is a lot of protein beyond the acceptable range that's recommended in humans. So we've got this range of protein intake, and then within this range of protein intake, we also have different levels of carbohydrate and fat. So within our 7% proteins, we have a low carbohydrate diet and a high carbohydrate diet, and a high fat diet and a low fat diet. So because these all sum up to the same level of energy intake, we can look at these on a kind of right-angled nutrition triangle, and we can see where all of these diets lie effectively within nutritional space. So we can see that this diet here represents diet 2, which has got 7% protein, 78% carbohydrate, and 15% fat. So this is that diet there. We've then got 3 other diets that also have 15% of their energy coming from fat, but they have a different protein and carbohydrate makeup. So this diet has 21% protein and 64% fat. This is actually the normal kind of macronutrient ratio that we would use in mouse studies. So in a normal mouse study, normal chow mouse study, we would use this diet 5, which is 21% protein, energy coming 21% of your energy is coming from protein, 64% from carbohydrate, 15% from fat. So we can basically use this model to systematically test whether the intake of different macronutrients can influence a specific health outcome. And there have been a number of high-profile studies published looking at the chronic effects of these diets on lifespan in mice. That's where I really first heard about this research, how it can influence other mouse models of disease, like mouse models of PCOS, and also chronically influence metabolism when you take these diets in for a long period. So you can basically treat mice with these 10 different diets. We can then look at a particular phenotype of interest. So we'll be looking at glucose tolerance, for example. So we measure glucose tolerance in all of the animals on these different diets. And then we can conduct a modeling procedure called creating mixture models, where we look at all 3 of these macronutrient percentages as predictors and as proportions, contributing to total energy intake. We then fit a series of different models with, a linear effect of protein, carbohydrate, and fat. And then we can also fit non-linear and interactive models as well. We then select the best fitting model for our data. And that could also be a null model where there's actually no predictive effect of diet. So we also include a null model, and we check that inclusion of these different factors improves the fit of our data. We can then create a heat map across this triangle, so we can look at our particular trait across this sort of space. So for an example, here is a heat map created for a particular trait. I'll actually show you this later in the talk. And what we can see is that for this particular trait, it's influenced by protein intake. So the color goes from blue to red. So this is a high level for that particular trait. This is an aspect of placenta morphology. This is a low level for that particular trait. And we can see that the color just goes uniformly along with the x-axis, which is protein intake. So you can see the more protein that the animal is given, the higher the value for this particular trait. If we look at along one level of protein, we can see that substituting fat for carbohydrate doesn't actually influence our trait outcome. So this is predominantly influenced by protein. We can then look across an equivalent level of fat. So imagine we're looking at these three 15% fat diets. We can see that as animals eat more protein for carbohydrate, that trait value increases. So if this was an in-person talk and I could see your faces, hopefully you would be nodding along now so that you understand the general process. Okay, so the first experiment we did, we conducted a relatively simple experiment where we took mice, C57, thanks for the thumbs up, C57 B6 mice, which is one of the main mouse strains that we use for biomedical research. We put them onto these 10 different diets. We left them on these diets for a week prior to mating, just for them to habituate to these diets. Half of the animals were then mated and allowed to become pregnant, and the other half were left with another female mouse and were kept as virgins. We left these animals until gestational day 17.5. So this is late pregnancy in a mouse. Mice usually give birth around gestational day 21, gestational day 20. And over this period, we sampled them for body weight changes. We conducted a glucose tolerance test at late pregnancy. We took a blood sample to look at their circulating insulin levels. We also dissected them. We euthanized the animals. We dissected their pancreas so we could actually look at how their beta cell abundance changed during pregnancy. And then we looked at fetal viability, growth, and placenta morphology. So we also wanted to understand, do these diets influence maternal health, but also do they influence the growth of the offspring? So obviously, any intervention that we develop for humans, we need to be really confident that it's not going to have a deleterious effect on the offspring. Okay, so I'm going to jump straight into the results. So one of the most striking things we found in this study is that high protein intake, specifically during pregnancy, impairs glucose tolerance in mice. So in mice, we can take our animals, we can fast them, inject them with a bolus of glucose, and then we can look at how that glucose level is taken up from their blood. So obviously we inject them, the glucose level rises, and then the glucose level goes back down again as tissues take up that glucose from the bloodstream. What we found is that in non-pregnant animals, there wasn't really an effect of protein intake on glucose tolerance in female mice. But during pregnancy, mothers seem to develop some real sensitivity to protein, such that mothers who are on high protein diets had impaired glucose tolerance. So in this figure on the left here, I've just split the diets into those that are low in protein or high in protein, so I can show you the glucose tolerance curve. And you can see that those females who are on diets that are high in protein are less able to take up this glucose from their bloodstream. By contrast, the control animals who are on the high protein diets have no problem with this. So what you can see is that actually this is kind of a linear relationship across these different levels of protein. So for the pregnant animals, which are colored in red here, the more protein they are eating, the more impaired their glucose tolerance is. So this plot here on the right-hand side is the area under the curve, and I'll be showing you that a couple of times in this talk. So a higher area under the curve indicates impaired glucose tolerance. So we can also make one of these heat maps. So this is a heat map for that glucose tolerance data. So we can see that animals who are eating more protein have a higher area under the curve, so more impaired glucose tolerance. And we can look at that on a number of different levels for a given level of fat. So we can imagine the three 15% fat diets here. The more protein that they're eating relative to carbohydrates, the more impaired their glucose tolerance is. We can also look at a given level of carbohydrate. So just go across, the more protein the mothers are eating relative to fat, the more impaired their glucose tolerance is. Now you can see that these lines aren't just purely vertical, they are diagonal, because there's also an additional effect of fat intake as well on glucose tolerance. So for a given level of protein, we also find that eating more fat relative to carbohydrate also impairs glucose tolerance. So we find that both increased protein intake and fat intake impairs glucose tolerance during pregnancy. So here are the I've just split the data so that we're just looking at pregnant low protein females. So effectively this plot here, and now I've split it according to the fat level. And what you can see is that females who are eating above the 15% fat level also have impaired glucose tolerance. These effects are actually quite small because it doesn't anywhere near match the effects we get of protein, but it's still a significant predictor in our model. Here you can see the heat map for control females relative to pregnant females, and they both use the same color scale. And what you can see is that in the control animals, the virgins, we're not really detecting an effect of diet, but in pregnant animals, we're seeing really strong effects of diet. So there seems to be some increased sensitivity to the metabolic effects of macronutrients that arises specifically during pregnancy in mice. And that also does seem to match up with the epidemiological associations that have been seen in humans. So we also looked at fasting insulin levels in our animals at late pregnancy. And we also found that in pregnancy, animals who are eating high levels of protein have higher fasting insulin levels, which is indicative of impaired insulin sensitivity. So here I've just converted the data so that we're looking at an individual level, how much protein each animal is eating. In the virgins, we don't see a relationship between protein consumption and plasma insulin levels. But in pregnancy, those animals that are eating the highest amounts of protein, and these are the ones on the high protein diets, they have higher plasma insulin levels. You don't see an effect of fat for plasma insulin. In fact, you see these graphs look slightly different because there's instead an effect of carbohydrate. So at a given level of protein, we can see this in both control and pregnant animals. Animals who have higher carbohydrate levels in their diet have higher plasma insulin levels. But this effect is exactly the same across control and pregnant animals. This probably represents the fact that we fasted these animals, but they probably still have some residual carbohydrates being digested when we took the blood sample. So we dissected the animals at late pregnancy, and we also looked at effects on fetal development. So we looked at the total number of fetuses, and we also looked at fetal viability. So obviously, mice are very different than humans. Instead of having one, maybe two babies, mice will have anywhere between sort of 6 to 11 or 12 fetuses within a pregnancy. And what we found is that while high protein intake impaired maternal glucose tolerance, it did increase fetal viability. So there's a trade-off between maternal glucose tolerance and fetal viability that's mediated by protein intake. So you can see here when we dissected the animals, the mice who were consuming more protein had fewer dead embryos or reabsorbing dead fetuses or reabsorbing embryos during the dissection procedure. And then we can convert that to fetal viability. This is mainly driven by the animals on the low protein diet. So on low protein diets, you can see quite a relatively low rate of fetal viability. So this is something that we need to be aware of when considering any manipulation that might alter protein intake during pregnancy. But really, we're seeing this effect at the lower protein levels. So we also dissected the animals, we took the fetuses, and we also took the placentas as well. We also wanted to look at placenta size and placenta morphology to see how this was influenced by maternal diet. So this is a cross-section through a mouse placenta, and we have two main regions within the mouse placenta. We have the junctional zone, which is where hormones are produced by these fetal tissues that influence maternal physiology, and then we have the labyrinthine zone where actual nutrient exchange takes place. So we can quantify the size of these and see how it relates to macronutrient intake. And what we found is a really striking effect of protein intake on the relative size of the labyrinthine versus the junctional zone size. So there's actually no overall difference in the size of the placenta, but the size of these two different zones changed quite dramatically with protein intake. So with greater protein intake, there was an increase in the size of the junctional zone, the zone that produces hormones and does various other things, but a reduction in the size of the labyrinthine zone. So the placenta is changing quite dramatically with these different diets. So here we can see this heat map for junctional zone size, and you can see it's predominantly influenced by protein. So as animals are having more protein in their diet, the junctional zone is increasing in size. So we can see this is going like that. Whereas the labyrinthine zone is decreasing in size with maternal protein intake. It's also decreasing in size in relation to fat intake too. So I'm going to push through quite quickly because I've realized I'm using up quite a bit of time. We also looked at placental lactogens as well, which are one of the hormones that are produced by the placenta that influence maternal physiology. And quite surprisingly to me, we actually found higher levels of these placental lactogens on low protein diets and high protein diets as well, which was potentially not what I was predicting. So anyway, we're seeing really striking effects of these diets on maternal glucose tolerance and circulating insulin levels, and also big effects on the morphology of the placenta. But from these results, it's really hard to kind of understand what the long-term impacts of these changes in placentation might have for offspring health outcomes. So we can see that maternal diet is influencing development, but we can't understand what the implications of those changes might be for the long-term health of offspring. We know from really striking human studies that undernutrition can be associated with impaired offspring health. The classic examples of the Dutch famine, where undernutrition at particular periods of pregnancy have long-term impacts on the metabolic health of those children. There's also some evidence that maternal intake of specific macronutrients in the context of overnutrition might also have health outcomes on offspring too. However, up until recently, most people have looked at the effects of maternal obesity on the long-term health of offspring. However, a paper was published earlier this year by some of our collaborators in Australia, where they looked at a Danish birth cohort and tried to understand how intake of specific macronutrients might influence adiposity of offspring. So they looked across 66,000 pregnancies where they had information on dietary intake of different macronutrients at gestational week 25. They then compared macronutrient intake, so they broke those dietary records down into a maternal intake of different macronutrients as a proportion of total energy intake. And they compared that to both maternal body weight and particularly postpartum weight maintenance, and also child adiposity too. And one of the striking things they found is that mothers who were eating high levels of protein, their offspring had the children from those pregnancies had a higher BMI at 11 years of age and also 14 years of age. So again, we've got a heat map, one of these heat maps here. And for mothers who are eating more protein, you see an increase in offspring BMI. These triangles here actually represent the acceptable macronutrient distributions for the USA and Australia. So the kind of recommended and perceived as acceptable levels of protein intake. And you can see that this variation is going across that level of acceptable macronutrient distribution. So there is various bits of evidence that maternal diet can influence the long-term health of offspring. But again, it's hard to understand from these epidemiological and human studies the causal impacts of different macronutrients on these health outcomes. So we've recently conducted another study where we have looked at the long-term consequences of these diets during pregnancy for the metabolic health of offspring. So this is an experiment that we've just conducted. It just finished a couple of months ago. It was run by a PhD student in my lab called Cameron Young. And we took female mice We placed them on these 10 different diets, again, 1 week pre-pregnancy. We then allowed them to mate, and they stayed on these diets during pregnancy. And then at birth, as soon as the offspring were born, we switched their diet so that they all went onto a standard, effectively diet 5, normal diet from birth to weaning. So during lactation, they were on normal diets. We also standardized litter size at this point. I was quite nervous that the mothers might lose their offspring at this point, because mice can be quite sensitive to environmental changes, but actually all of the mothers kept their offspring. So we had 6 females per diet, and then we weaned 2 to 4 offspring of each sex from these mothers. So we had 210 offspring weaned. We then allowed them to develop to 100 days post-birth, which is really full adulthood for a mouse. We looked at their body weight over this period. We've looked at their body composition, their glucose tolerance. I'll show you some of those results today. We're also going to look at their circulating insulin levels, their pancreas abundance. We conducted echocardiography on these mice. We're going to do cardiac fibrosis and also conduct metabolomics and epigenetics on these samples. So I'm just going to show you a couple of bits of data from this study. So we took the mice, we kept them on these diets from mating, so from the start of pregnancy to the end of pregnancy, and then we switched them to a standard diet at birth. They then stayed on this standard diet until weaning, and it was at weaning that we weighed these offspring and took them away from their mother. What we find in terms of their body weight at weaning is that there are some effects of this dietary manipulation during pregnancy on body weight at weaning, which is most strongly found on this low protein, 7% protein diet, particularly where it's matched up with low fat. So offspring from mothers who are on low protein, low fat diets had a lower weaning weight than the other groups. So you can see this on the heat map here, but just to kind of familiarize you with this figure, I've also just got the means and error bars of each of these 10 individual groups here. So this is the 7% protein diet with low fat. This is the 7% protein diet with high fat. You can see there's definitely an interaction between the level of protein and the level of fat to carbohydrate ratio within this diet as well. So there's an effect of protein on weaning weight, and there's also an effect of fat as well. So mothers who are eating beyond the normal 15% fat intake, their offspring had a higher weaning weight, and mothers who are on this low protein diet, the 7% protein diet, also had a lower weaning weight. And then weight was about normal across the other diets when you controlled for this continuous effect of fat. So we then allowed the offspring to go from weaning, which is 21 days of age, up until 100 days of age. And then we weighed them regularly over this period. And what we observed, particularly in males, is that animals who were on the low protein, low fat, high carbohydrate diets maintain this lower body weight throughout their life or up until adulthood. So we still see that these animals here have a lower body weight, but we also see a suppressed weight in offspring from mothers who are eating high protein diets as well. So although these animals were weaned at a normal weight, they showed a depressed body weight by adulthood. So they were less able to grow across that post-weaning developmental period. So there's some consequence of high protein intake that suppresses growth in at least in male mice. In female mice, we actually don't see the same relationship. So we actually, when we do our statistical test, we don't find that any of the macronutrients predict body weight in the females. I can still make the heat map, and you can still see a slight color change across protein levels, but actually the absolute difference in body weight here is really small. So it's only like 0.5 gram. And macronutrient intake does not predict body weight in female mice. So in the females, which have a smaller body weight relative to the males, so males are averaging about a 30-gram body weight. In adulthood, females are averaging about a 23-gram body weight. Females with their lower growth rate are able to kind of compensate for those differences those early life differences, but it's the males which have higher growth rates, which seem to be showing the stronger effects of maternal dietary intake. We also looked at body composition of animals in adulthood at 100 days of age. And while females didn't differ in total body weight, they did differ in body composition. In particular, we find that females who are from mothers who ate high levels of protein and low levels of carbohydrate have greater adiposity. So they have effectively the equivalent of a greater BMI compared to animals who were from high carbohydrate, low protein diets. So there's some long-term consequence of maternal protein intake or maternal diet for adiposity of females. When we look at the males, the data is obviously more complicated by the fact that we see this difference in body weight. And actually this difference in body weight here is mainly driven by lean mass. And if someone wants to ask me at the end of the talk, I can show you those figures as well. But we also find an effect of carbohydrate on male adiposity. Now, all of these things are kind of co-correlated. So these animals here who are on the low carbohydrate, high protein, medium fat diets have the highest adiposity. So we're finding some effect of maternal diet on body composition and adiposity as well. So we've conducted glucose tolerance tests on these animals at 100 days of age to see whether maternal diet is actually influencing the metabolic health of offspring in adults. And here, as we may have predicted, we see a non-linear relationship. So we find that, at least in male mice whose mothers ate either low protein or high protein have impaired glucose tolerance. And indeed, people have shown this effect of low protein before, but we're also seeing this effect of high protein too. So here, just to show you the actual data, I'm just showing you, because it gets really messy if we show 8 different protein curves, the curve for our animals on the normal standard protein diet, the low protein diet, and the high protein diet. And you can see that both high and low maternal protein intake has an impact on glucose tolerance in male mice. So either abnormally low or abnormally high protein intake can both impair glucose tolerance. We also see the same relationship in females, although it's weaker than the relationship we see in males. Okay, so that's the main set of data from our preclinical work that I want to talk to you about today. And our key findings from this is that high protein diets and also high fat diets, even when we control and normalize differences in energy intake, can impair maternal glucose tolerance. And this is actually consistent with the epidemiological associations we see in humans. We also see this effect of maternal protein intake on offspring metabolic health. And here we see that both low protein intake and high protein intake can influence glucose tolerance. So we're really just scratching the surface with this data set, and we'll understand a lot more about effects on cardiovascular function and things like that as we get through the other aspects of the data. So in terms of moving forward in a preclinical sense, I'm really interested in understanding whether there are specific amino acids that are involved in causing these effects on maternal glucose tolerance. So I hypothesize, and indeed other people have hypothesized, that the effects of high protein intake may be driven by specific amino acids, in particular branched chain amino acids, so leucine, isoleucine, and valine. So these are the amino acids that we typically see increased in people who eat a lot of protein or that have obesity. And we know that these branched chain amino acids can reduce insulin sensitivity by acting on particular signaling pathways in tissues. So I would like to conduct an experiment where we actually manipulate the abundance of specific amino acids, and then we can experimentally test whether mothers are sensitive to specific amino acids during pregnancy. I'm also interested in understanding how maternal diet influences offspring health. So we know that there's been research done on the effect of low protein diets relative to normal diets on offspring health. And it's been shown that low protein diets during pregnancy can impair pancreas development. So I expect that the results we're seeing for low protein diets will be a consequence of that impairment in pancreas development. Are we seeing the same thing with high protein diets, or is there some other physiological change that's driving those changes in glucose tolerance? We might be seeing some kind of epigenetic changes in tissues also. Okay, so I'll spend the last few minutes, and sorry, I've talked quite a lot about the preclinical research, about next steps for translation and where I'm thinking about going. So I've spent some time at the Emory Global Diabetes Research Centre talking to different researchers, and I'm really appreciative of the time I've had here. And actually giving this talk gives me a chance to reflect on the different conversations I've had and to think about what the next steps for translation might be. So it's been suggested to me I need to understand what are the actual observed range of protein intake we see within New Zealand, and that will help us understand how potentially important these effects might be, and how does that correspond to the recommended range of protein intake. We also need to think about do levels of protein intake we see within different populations differ by ethnicity or in at-risk groups. We then need to think about what might be a suitable intervention when we weigh up all of these different factors in terms of maternal health and offspring health. What might be a suitable intervention generally? Do we already know what intervention might be? And might we be able to adapt that for New Zealand? Or do we need to think about interventions that might be beneficial specifically for at-risk populations in New Zealand? It's also been highlighted to me that I need to think about what evidence would be required to convince people to update their guidelines, change their behaviors, or develop an intervention. Do I have enough information now? Sorry, there's a chat comment here. That's just from Wendy. What have I got enough information to convince people that this is important clinically, or do we need further follow-up studies? So actually, I've looked since I've spoken to people at what the observed range of protein intake is within New Zealand. So there have been New Zealand adult nutrition surveys. The mean for women of reproductive age is 17% of energy is coming from protein. So, that's like within that range we were looking at in the observational studies. But in terms of looking at the 90th percentile, if we look at the 90th percentile, these women are taking in 24% of their daily energy intake from protein. So this is really a really high level, and it might be specific groups of people who are taking high protein that we might need to focus on when thinking about interventions. In terms of recommended and acceptable ranges, the recommended range for humans is 10 to 20%, but the acceptable range, according to the World Health Organization, is actually 10 to 35% of your energy coming from protein. So I would predict that people who are eating these higher levels would really be at risk of metabolic dysfunction. So if we think about dietary recommendations for New Zealand and what women or pregnant people are recommended to do during pregnancy, well, actually, currently, we recommend women to increase their intake of food groups that are high in protein. So, at the moment, what's emphasized is increasing protein intake for the healthy growth of the offspring. So here we can see that pregnant women are recommended to eat, according to this guideline, 30% more of these protein-heavy foods than women who are non-reproductive. And the only other thing we're recommending women eat more of is these grain-related foods. In terms of changes in diet in response to gestational diabetes, what are the actual recommendations we have when women develop gestational diabetes? Well, these are largely the same as for type 2 diabetes. We don't have a specific set of guidelines tailored for pregnant people. We expect promotion of high quality nutrient-dense carbohydrates, an increase in unsaturated fats relative to saturated fats, but specific intervention studies that are focused on dietary changes to reduce gestational diabetes have largely failed to show benefits of specific diets. So we need to think about how this might be adapted. So we are in New Zealand have been thinking for a long time about the relationship between diet and metabolic health in pregnant women. So I'm part of a longitudinal cohort study looking at energy changes in women. So we're conducting a longitudinal study where we're recruiting 70 women who plan to be pregnant. and 70 women who don't plan to be pregnant. We're looking at changes in their metabolism and also in their sensory perception and food and preferences for different foods. So this was based on earlier work that we'd done, showing that there are long-term changes of motherhood on adiposity and body weight. We can also bring people into the lab and we can look at their preferences for different macronutrient foods. So we can bring them in, we can give them a buffet, and we can look at whether women prefer foods that are either high in protein or high in fat. Sorry, I'm going to wrap this up in a moment. So we're thinking about now moving a step forward with this and using continuous glucose monitors to assess diet responses during pregnancy. So we can look at both chronic differences, so women who are chronically eating high levels and low levels of protein, and look at their glucose levels across pregnancy, and also look at acute responses to different meals. I'll also highlight, and I will stop in 1 minute, but that in New Zealand, we also have to be aware of any of our interventions need to be concordant with a treaty that was signed between British settlers and Maori when New Zealand was first colonized, called the Treaty of Waitangi, which is still very much in operation today and protects Maori rights within New Zealand. So the government of New Zealand, according to this treaty, has effective stewardship of the health system, but it needs to be ensuring equity in health outcomes. And at the moment, we don't see that in New Zealand. Maori people still have a reduced life expectancy and increased risk of chronic disease. So we need to think, when we're developing these maternal health interventions, how they would be affected within the Maori community. So Maori have a different community structure to New Zealand Europeans. Family is really important. Maori whānau, they tend to spend more time in extended family groups with grandparents, have more children. An intervention that we develop, which is focused just on the mother, probably isn't going to be very effective and uptaken by the Maori community if you're ignoring the rest of the whānau. We also need to ensure appropriate governance and co-design in these interventions. And later on, I'm on sabbatical at the moment. I'm working with an iwi organization, a Maori tribe organization, to try and thinking about developing these interventions. So I'll leave it there. I'd like to thank the Emory Global Diabetes Research Centre for hosting me over the last few weeks. I've had a really good time. The Otago Medical Research Foundation for funding this work, and everybody in my lab, in particular Cameron Young and Faye Morland, who've led these projects. Thanks, everybody. Hopefully we've got some time for questions.
That was great. Thank you, Mike. We do have a question. Gabriella, if you want to go ahead and unmute yourself.
Hi, my name is Gabriella. I'm an undergraduate researcher at Emory in my final year at the moment. I really enjoyed your presentation, so thank you so much for taking the time to come and speak to us today. I'm really interested in intergenerational health, so this has been great. I wondered, because this project is centered around the maternal influence, how you had isolated the paternal effect from the results, whether you had controlled the paternal diet as well before mating, or if there was any post-control analyses, particularly because of, I know you mentioned that the placental ratio between the junctional versus labyrinth tissues was one of the notable results.
Yeah, thank you, Gabriella. That's a really good question. So I'll just go back to our design. So the mothers were on the diets for a week before mating, and then we basically put the males in, and I would say about half of our females mated with the males within 1 to 2 days. So for any of these diets to have had an effect on the father, it would have had to have happened really quickly. So I don't think it's realistic for the fathers to eat these diets to cause a change in sperm epigenetics or something. However, I would say that there's a paper published in Nature Communications last year, Crean et al. 2024, that has done this experimental design, but looking at effects on the father. And they found big effects of macronutrients on the health of offspring as well, and different ratios compared to what we've seen. So yeah, there's definitely going to be a paternal influence as well. And when I get some headspace, I really want to map the two different sets of macronutrients, because I think in their study, they found much stronger effects of carbohydrates or paternal carbohydrate intake on offspring health outcomes.
Wow, that's really interesting. Okay, thank you.
Thank you.
Thank you for that talk. I'll ask a question while we're waiting for others. So, in human populations, we have to think a lot about pre-existing metabolic morbidities as a risk factor for metabolic health during pregnancy and beyond. I know in the mouse models that you're working with, you are able to achieve experimental conditions and comparative conditions across those mice, but what role does that pre-existing metabolic morbidity in women and in female mice, how does that translate, and how would you connect those pieces? So how applicable are these findings then in the current times where we might not be starting from a position of metabolic health?
Yeah, no, I think that's a really good point. And lots of people have done research on obesity in mice. So, we want to actually partition the effects that are not due to obesity here, but there are various different studies that have used high fat, high sugar, high calorie diets to get mice fat and then look at the health outcomes for the mother and the offspring. The effect on glucose tolerance that we see actually aren't that striking so people have done these experiments, have got female mice fat, done a glucose tolerance test during pregnancy, and they don't see the same effect that we see in terms of protein intake. But I think that's definitely a whole separate thing that we need to understand. And I guess that was something that we were trying to actually avoid as a confounding factor. I did want to redo this experiment, and I applied for money to redo this experiment where we have another factor which we tie into it, which is energy intake. So we've got those 10 diets, and you can also vary within those 10 diets the density of all of the macronutrients. So you can then look whether energy intake across those different levels of macronutrients. You can use 30 diets, and people have done this for a lifespan study. And then you can see, is the effect of protein, for example, stronger than the effect of energy intake and obesity? But we haven't been able to do that yet. It's quite a big undertaking, and it's quite hard to convince people that the full design is worthwhile.
Thanks. Any other questions? Yes. Seeing none, thank you so much for this provocative presentation. I think everyone knows how to reach you based on the flyer. And thank you very much for joining us. And thanks to the audience as well for showing up on a hot July day.
There is a quick question.
Another question, sorry.
Yeah, someone's just come up with a question.
Yep, let's go.
Sorry, I put it in the chat. This is Chase here. Thank you very much for the presentation. I'm in a noisy background, but I just put a question in the chat if you have a minute to answer.
You showed a study timeline figure later in your presentation showing when the experiment diet was begun. Do you think it's worthwhile exploring dynamic diets throughout gestation, or do you think there is a fixed optimal macronutrient balance that exists for the duration of gestation? No, I think you are right. And particularly when we're thinking about outcomes for offspring, and this is an experiment I would like to do, particularly in relation to hunger and obesity. So people have looked at effects of high fat diets during specific periods of gestation. And, during specific periods of gestation and lactation, the fetal brain is developing. So there may be periods which are particularly sensitive to the effects of macronutrients, particularly for offspring health outcomes. So, I would predict that maybe high protein diets have an effect on offspring physiology that occurs during a specific developmental window during development. Now, I would say that fetal developmental window in the mouse is probably not going to map onto the human because developmental rates are different. But yeah, it's certainly something that's worth exploring and something that I am thinking about for future studies.
Yeah, thank you. Thanks, sir. Yeah, I would imagine that that's probably a much more complicated, time-consuming experiment. You might need a larger sample sizes, more combinations. But no, yeah, thanks.
Yeah, I would really like to do it. And in mice, they're born much, they're still very underdeveloped when they're born. So actually, some of that neurological development also occurs in the start of lactation as well. So we might actually want to do these experiments going across pregnancy and lactation. And indeed, we have done some experiments looking at that, but mainly focusing on maternal health rather than the health of the offspring. Thanks, Chase. Sorry, Shivani.
No, that was great. Any other questions? All right, then. Well, thank you very much.
Thanks, everyone.